Hello, my name is Amaan Siddiqui, and I'm a senior at Johns Hopkins University. I'm an anthropology major, a pre-medical student, and an otolaryngology research assistant in the lab of Dr. Amanda Lauer. My poster is titled Histology of Macaque Models as a Pedagogical Tool for Cochlear Histology and Anatomy. Taking a look at the abstract on the left, this project aims to provide students, scientists, and physicians with a deeper understanding of cochlear histology and anatomy using the image analysis of a rhesus macaque model. This is significant because macaque models have a complete temporal bone and cochlear size and shape that closely resemble a human's cochlea. Macaque models also appear to more closely resemble humans than other models like mice, which are frequently used to study hearing. Additionally, it is incredibly difficult to obtain pristine, intact human cochlear tissue because the fixation occurs post-mortem and most donors are of older ages. As such, histological image analysis of a macaque temporal bone structures will provide an effective way of learning the anatomy and physiology of hearing. In our lab, we study hearing loss through the histopathology of temporal bones. In order to better understand said hearing loss, it is imperative to understand the normal histology and anatomy of the cochlea and the functions of its manifold cells, organs, and membranes in order to train researchers to gain expertise in histopathology. We can use high-quality tissue specimens from macaques to develop analysis protocols using artificial intelligence techniques, semi-automated techniques, or manual techniques, all of which can be translated to human ears. Looking below at the objective of this poster is to demonstrate the effectiveness of macaque temporal bone models for teaching cochlear histology and anatomy. Directly below the objective, the methods section is listed, and the methods will be discussed in depth at the live presentation of this poster at the Spring 2023 Our Dreams Research Symposium. Looking upward at the results, Figure 1 shows a slide scan image of a macaque temporal bone section on a horizontally cut plane. The vestibular system is also visible here. Figure 2 has been magnified to focus on the entire horizontally cut cochlea and its spiral ganglion neurons, which have been labeled in the Rosenthal's canal. Figure 3 is magnified to focus on a single turn at the bottom right of the cochlea, as well as its scala media, scala tympani, and scala vestibuli, all of which have been labeled. Figure 4 is zoomed in even more to focus on the Reisner's membrane and the stria vascularis, and Figure 5 has been zoomed in even closer to focus on the organ of Corti and its surrounding structures, all of which have been labeled on the anatomical key below, which will also be discussed in depth at the live presentation for this poster, alongside an explanation of each of their functions. If you look at Figure 6, you'll see an image of a rhesus macaque whose species is being studied in this poster. Taking a look at the conclusions, in our experience, using this model of a normal macaque temporal bone, we found that the structures closely resemble a human temporal bone. Using this model as an effective teaching tool for providing the necessary analytical skills related to cochlear and vestibular histology and anatomy. With this understanding of normal histology, we can work on developing other macaque models to study otopathology such as Meniere's disease, otosclerosis, and other conditions. For future directions, our lab will be using this teaching tool on a temporal bone website that we currently have under development, as well as use it for training new lab members. We will develop a similar teaching tool for studying vestibular structures and balance. If you have any additional questions, feel free to use the QR code for the Lauer Lab website. Reach out to me, Dr. Balhi, or Dr. Lauer. Have a great day, and thank you for listening.